In this video, we are going to look at the process of blood coagulation, how the body produces a blood clot. First, we need to familiarize ourselves with a few basic terms. The first term is hemostasis. Hemostasis is the process of stopping blood loss. The main mechanism for how this is accomplished is blood coagulation. Fibrinogen is a plasma protein that gives rise to fibrin. Fibrinogen is an inactive protein, always found in the plasma. When it's needed, pieces of fibrinogen break off, leaving behind fibrin. A blood clot is this red gelatinous mass made up of a mesh of protein filaments, which is the fibrin, and various trapped formed elements of the blood. Now, blood clots can only form in veins, capillaries, and small arterioles. The blood pressure is too high in larger arteries, and this will prevent them from forming. As soon as they would form, the pressure in these vessels would blow the clot apart. And then there's thrombin. Thrombin is an enzyme. It is derived from an inactive precursor known as prothrombin. So we see here prothrombin will be activated into thrombin. Once we have thrombin, thrombin is responsible for splitting fibrinogen to form fibrin. Then once we have fibrin, these strands of fibrin help to stabilize our blood clot. Thrombin also activates clotting factor number 13, takes it from an inactive form to an active form. Clotting factor 13 also helps to stabilize a clot by stabilizing fibrin. It will form crosslinks in the fibrin molecule, which helps to stabilize the clot. The coagulation cascade represents a series of several enzyme-activated steps that ultimately lead to the formation of fibrin. This clotting cascade involves various clotting factors. These clotting factors all circulate in the blood in an inactive form, so they are always present. They will only be activated when they are needed. Once needed, the first in the series will be activated, and then this in turn activates the next, and so on. You need to be aware of the various clotting factors and what their functions are. And this list here shows the clotting factors. We have already talked about fibrinogen and prothrombin, as well as clotting factor number 13. We will be mentioning a number of these others as we go through. Now, what triggers or initiates this clotting cascade? There are two basic mechanisms. The first mechanism is called the extrinsic pathway. Extrinsic, external, because all of the factors involved are found external to the blood in order to get this cascade started. The other pathway is the intrinsic pathway. Intrinsic or internal, all factors necessary to initiate the cascade are present in the blood. We'll start first by looking at the extrinsic pathway. Now, this looks like a complicated pathway. You are not responsible for all the various steps in here. The extrinsic pathway, as we just said, the initiating factors are outside of the blood. So what starts this pathway is some sort of tissue damage. When tissue is damaged, it results in the release of a material called thromboplastin, which is also known as tissue factor. This is exposed whenever tissue is damaged. Tissue factor then activates clotting factor number 7, takes it from the inactive form to the active form. Once we have clotting factor 7, the important thing that it does is activate clotting factor number 10, take it from its inactive form to the active form. Notice what clotting factor 10 is responsible for. This is what takes prothrombin to thrombin, and we're back at what we initially started talking about. The second pathway is the intrinsic pathway. Again, there's a lot more steps on here than you are going to be responsible for. Intrinsic means it is started internal, or everything is inside the blood vessel. So here, what starts this pathway, when blood vessels are damaged, it will expose and damage the endothelial surfaces. Blood vessels are lined with a single layer of endothelium. Right underneath that endothelium is collagen. When this endothelial surface is damaged, this will expose that collagen. So here it started by damage to the endothelial surface, and we skip through all these steps here. We see that the intrinsic pathway also is responsible for activating clotting factor number 10. And then once we have 10, this is what takes prothrombin to thrombin. Both pathways start out differently, but they both result in the activation of clotting factor 10. So to make this chart here complete, factor 10 would go right here between prothrombin and thrombin. After clotting factor 10, both pathways are exactly the same, resulting in the activation of thrombin, which then activates fibrin. Both of these pathways also need small amounts of calcium. Calcium is actually known as clotting factor number 4. Calcium helps to activate several steps in this cascade. Many times when we take a blood sample, we do not want the blood to clot. Maybe we're measuring clotting time, prothrombin time. So when you draw the blood, in the tube will be a special material called a chelating agent. This chelating agent will prevent the blood from coagulating because it removes the calcium. All you have to do in this clotting cascade is remove just one of the factors and you will not get a clot. This is very easily reversed when you want the sample to clot. Just add back a little bit of calcium and you can run your tests. Another important factor in forming a clot is a platelet. So we're going to look at the role of the platelet. Before we talk about this, I want to explain this picture. This picture represents the ultimate end product of a clot. Down here at the bottom, these brownish strands, these represent collagen. Remember I said a blood vessel is lined with endothelium. Right under the endothelium is collagen. When that endothelial surface is damaged, this will expose that collagen. These structures represent the platelets, and these gray strands represent the fibrin. Now, when there is damage, blood will be flowing to the site of damage, and as the blood flows, the platelets will become trapped. One of the clotting factors, known as clotting factor 8, binds to a material called von Hildebrand's factor. Now, clotting factor 8 is not represented on this picture, but von Hildebrand is. These are the triangles that we see on the collagen. Von Hildebrand is bound to the collagen. Platelets have receptors for von Hildebrand. So when there is endothelial damage and the collagen is exposed, so too will the von Hildebrands. Since the platelets have receptors for this material, as they travel by this damaged area, they will become attached to the von Hildebrand, and once they are trapped, the platelets become activated. They will change. We're going to look at the various steps in platelet activation. Once platelets are activated, we will see this accumulation of more platelets at the site of damage. This promotes coagulation or the formation of a clot. Also, this entire process, as it continues, will be controlled. And we're going to look at how this all happens. All right, the steps in platelet activation. There are 4 steps involved in activating platelets. The first step is the formation of a platelet plug. So again, to recap, the blood vessel wall was damaged and damaged the endothelial lining. This exposes the collagen with the von Hildebrand's factor. The initial platelets arrive at that site of damage and become bound to the VWF. Once they're bound, their shape changes. They become much rounder with numerous projections. Once this happens, they are referred to as an activated platelet. These activated platelets are now able to bind other activated platelets. More and more platelets will begin to stick, and as they stick, they too become activated. This process will continue, will be repeated until we have a plug. A plug is nothing more than a mass of platelets. It will provide a temporary stop to blood loss, but it is not very stable, not very strong, so we need to reinforce this. The second step in platelet activation is called the platelet release reaction. In general, what's going to happen in this step is we will see the release of various substances that will help to control the response. These substances actually amplify the overall response so that you will see a very rapid formation as well as consolidation of this platelet mass. All of these substances are found within the platelets. They are stored in vesicles, and they will not be released until the platelets have been activated. This process of release is known as degranulation. All of these materials will be released from the platelets via exocytosis. This represents some of these factors and what their role is. The first is ADP. ADP, follow the line around, ADP promotes platelet activation. So it's going to stimulate this process. Another material is thromboxane A2. Follow this around. It too promotes platelet activation, platelet aggregation, and the more release of these factors. Thromboxane A2 also is responsible for vasoconstriction. What this will do is close the gap at the site of damage so that you will ultimately need a smaller clot in order to stop the bleeding. Both of these substances, you see here, both of these substances also react with thrombin and will trigger clot retraction. This is a later step in the process, and we will come back to this. Another material that is released from the platelets are growth factors. Growth factors promote the movement of fibroblasts into the area of damage. These will play a role later on. We will have a whole unit devoted to the repair process. Our third step in platelet activation is the exposure of surface receptors. One of these factors is called platelet factor 3. Platelet factor 3, these are the upside down U's that are shown here in this picture. Platelet factor 3 will bind clotting factor 8 and 5. 8 is not shown, but clotting factor 5 is. This is what looks like the letter Y. This helps to catalyze the formation of fibrin and localize it at the site of platelet aggregation. Basically what it does is helps to confine the response to where it is needed. Now notice this step is called exposure of surface receptors. These receptors now appear on the surface of the platelets. So here again is platelet factor 3, which will hold on to bind clotting factor number 5, which helps to organize and localize these strands of fibrin at the clot site. So what this does, it also exposes receptors that are specific for fibrinogen and fibrin. These are, you can see them here, fibrinogen and fibrinogen receptors and a fibrin receptor. This will bring the fibrin into position so that factor 10 can activate the thrombin. This helps to anchor the platelets and to form a very localized mass of coagulated blood. The last step in platelet activation is clot retraction. As the process continues, fibrin will form this tangle of filaments that will be anchored to the surface of the plug. This also ends up trapping red blood cells as they pass through. This is what results in this loose gelatinous mass at the site of damage. We need to consolidate this mass. We need to make it thicker and stronger. So the next thing we see happening is that the platelets will contract. Since the fibrin is attached to the platelet, as they contract, they pull on these strands, making them very tight and condensing that gel. As a result, this will squeeze out a lot of the plasma and liquid, resulting in a very dense gel. Now we're going to look at a little bit more at thrombin and some of its other roles. It does more than just to convert fibrinogen into fibrin. One thing, and so this is the pathway that we've looked at already. Fibrin also helps to intensify the coagulation cascade. This is a positive feedback loop, ensuring that more and more thrombin is going to be produced so that more and more fibrin can be made. Thrombin also promotes platelet activation and the role of the platelets. This too will stimulate that cascade, resulting in more thrombin. Also a positive feedback loop. Now, all of these positive feedback loops need to be controlled. We want to prevent forming a clot that is larger than necessary. If a clot becomes too large, it runs the risk of preventing blood from getting through that vessel, and that will be very dangerous to the downstream tissues. They would not get their nutrients and oxygen supply. So there are three factors that help to maintain homeostasis, help us to regulate the overall size of a clot. These include hemodynamics, which is the basic flow of blood, endothelial mediation. This refers to normal endothelium on either side of the area of damage, and the fibrinolytic system. We will look at each one of these. First, hemodynamics, or the movement of blood. Many activated factors will be carried away by the blood before they have a chance to bind to the surface receptors or activate other factors. So essentially they're removed and flushed out of the system as the blood is passing through. Now you may say, well, can't they just travel through the body and come back and do their job? No, these activated factors, when they are removed, as they travel through the liver, they will be inactivated and hence unable to return to the site. Also, as new blood arrives into the site of damage, it will dilute out the existing factors. Both of these together help to slow down the cascade. The second factor is endothelial mediation. Again, this involves normal endothelium adjacent to the site of damage. Now here's another role of thrombin. Thrombin triggers normal endothelium to release a material called prostacyclin. Prostacyclin is a prostaglandin, and it helps to inhibit platelet aggregation. In this way, platelets cannot adhere to the neighboring normal surfaces of the blood vessel. They can only play a role where there is actual damage. Normal endothelium also produces enzymes that degrade both ADP and thrombin. Remember, ADP was one of the platelet release factors. If ADP is affected, you will see less platelet aggregation and activation. If thrombin is affected, it will interfere with the formation of fibrin. Normal endothelium also produces other enzymes that will break down the clotting factors. Normal endothelium have receptors that bind a material called antithrombin 3. Antithrombin 3 plus heparin, which is found in the plasma, will inactivate thrombin and other clotting factors. However, it only degrades those factors that are not already bound to the platelets. And lastly, when thrombin binds to a receptor on normal endothelium, it will activate a material called protein C. Protein C inhibits the coagulation cascade by inactivating both factors 5 and 8. Next, the last one is the fibrinolytic system. This provides a means for clot breakdown. This will allow for phagocytosis and cells to come in and start degrading the clot. The process also consists of a system of 2 separate cascades, resulting in the formation of plasmin. Plasmin is what's responsible for breaking down fibrinogen, fibrin, clotting factor 5, and clotting factor 8. Plasmin also comes from an inactive precursor called plasminogen. The fibrinolytic system. So there are 2 cascades. The first cascade involves the damaged endothelium. Again, when the endothelium is damaged, it results in the release of a material called tissue plasminogen activator, also known as TPA. TPA being the clot buster. TPA activates plasminogen, converting it into plasmin. TPA will affect the breakdown of existing clots. Plasminogen is actually bound to fibrin within the clot, and essentially it's built into the clot, just waiting for TPA. So this allows for the clot to be broken down from within. The second cascade involves subepithelial tissue factors, or these are known as other activators. These activate clotting factor 12, which then activates plasminogen. This cascade, this pathway helps to control clot formation and also allow for its breakdown. This entire process also needs to be controlled. Overreaction of the system is prevented by a material called alpha antiplasmin. We see this over here. One objection I have to this flowchart, alpha antiplasmin should actually be up here, because what it does, it inhibits the conversion of plasminogen into plasmin. It does not inhibit the action of plasmin. So this should be up in this area. So the overall goal is to strike a balance. The goal is to have enough coagulation to prevent blood loss, but limit it so that the flow through that vessel is not restricted. So how is this done? One is through the activation of clotting factor 12. It plays a role in activating the coagulation cascade, plus it activates the conversion of plasminogen to plasmin, so it controls the formation of clot as well as the breakdown of the clot. ADP, one of those platelet release factors, released by platelets to promote activation and coagulation. But ADP is also broken down by normal endothelial cells, and when this happens, it yields anticoagulant properties, so it helps to build a clot as well as control the size. And lastly, thrombin. We've seen a lot with thrombin in this unit. Thrombin activates platelets. It also causes fibrin production, but it also activates protein C that causes the release of TPA. When TPA will break down fibrin that the thrombin has formed. Thrombin also results in the secretion of PGI2. This will decrease platelet aggregation and activation. So again, it plays a significant role in the formation of a clot, but also helps to control the size of a clot.